Okay, everyone, we'll start for today's session. So, first of all, two questions like, can you all hear me and can you see my screen?
Yes, we can. We
Yeah,
can
okay.
hear you. We can see your screen. Thanks.
Yeah, sure. Thank you so much. So, yeah, good afternoon, Gupta and everyone. Week six of the course Research Methodology in Natural Sciences. And for those who are attending my TA session for the first time, I'm Arjun SB and I'm a second year PhD student at Indian Institute of Science, Bangalore. So, what we're going to do in this session is just going to be a repetition of the past week or the past 4 or 5 weeks, I would say. So, we'll go through a brief recap of all the lectures that have been taught in the week six of this course, and after that, we'll have a brief doubt session, and after that, we'll go on to discuss. Last week we haven't discussed week five's problems, so this week we will discuss week five's problems, and after that I'll discuss a few problems from week six, and then we will have another doubt session. Yeah, I think someone raised their hand in the middle. My okay. So one general instruction, as always, is that I would advise everyone to mute their audio as well as their video. This is just for um, the other audience in the call to not have any sort of disturbances while the lectures or discussion is going on. You can unmute yourself or you can raise your hand if you want to speak in the call. So with that being said, we'll jump right into today's session. Okay, so last week we saw about reproducibility, falsifiability, and Dr. Soumitra Banerjee us more about told us more about reproducibility, falsifiability, how to propose a hypothesis, and last week I think he started off with a discussion on statistical components or statistical measurement components such as population and samples and population means, sample means, and everything. So this week is sort of a continuation where from where we left off in the last week. So the this week how last week ended with the introduction of the normal distribution with the mean, median, mean and standard deviation, and how different types of standard deviations are used to assert a confidence of a certain percentage. So this week the lecture started with the topics of central limit theorem. So, and as usual, we got an introduction of into sample mean and population mean, and sample standard deviation and population standard deviation. So this week we jumped right into central limit theorem. So last week we had a last week kind of ended in an open-ended question as such, as in saying that does all distributions end up being normal? Does all distributions give us a sort of a Gaussian curve? Some distributions give us a very skewed curve. So what happens in that case? How do we resolve them, or how do we take any sort of data out of them? So central limit theorem starts off with that question and comes up with a theorem saying how this kind of problems are resolved. So central limit theorem in verbal form says that no matter like how skewed our population is, how much skewed our population is, and if we have a very large sample size, sample is nothing but a part of the population. So let's just say a population consists of hundreds and thousands or even in infinitely number of observations. A sample consists of a small amount of that population, let's just say 25 to 30 number of observations, which will help us study the whole population. So central limit theorem asserts that if the sample size is large enough, then the sampling distribution of the mean of the samples n. So if we take sample 30 samples out of a population, and then if we try to calculate the mean of all the samples, and then if we try to plot the mean, what we would get is essentially a normal distribution of the means. So the means eventually would form a normal distribution, which would have the mean same as that of the population mean. So it would have the same mean x bar, and it would have the standard deviation sigma by root of n. So sigma was the standard deviation of the population. So if we try to plot the means of all the data that we get from each sample, then the uh, standard deviation of the means, which is eventually called as standard error in the further lectures, that would become to be sigma by root of n, where n is the number of samples presented. So eventually, whatever skewed data we have gets resolved into a normal distribution. So the distribution of the sampling distribution will be normal if n is large enough. If the n is small enough, and in this lectures we consider a large sample to be somewhere around 25 samples. So if we have a small number of samples, let's just say um, 10 samples or 12 samples, then this theorem might not work out. Nikhil Sapre, sir, what is that called as? I'll come to that. I'll come to that in some while. Yeah. So now we have a brief idea of how to kind of study any type of data, whether it's a normal data or a skewed data. If it's a normal data, we can study as such. If it's a skewed data, we can sort of plot its mean distribution into a normal data and then study the data. And in further examples, like in the lectures also discussed some form of examples where we have some amount of population, where we are given some population mean and standard mean, and we are asked to calculate what kind of sample mean will lie beyond some range, or what kind of confidence interval do we need to calculate. So the normal sample size, the normal sample size if plotted in a curve, if plotted in a normal curve, and if the standard deviations are marked in such a way that the mean is there and the mean is bound by standard deviation in the negative range and the standard deviation in the positive range. So the area sort of bound by this curve gives us almost samples which gives us a confidence interval of about 63.7%, or if I'm not wrong, it's yes 63.7%. So that means that 63.7% of the population lie within one standard deviation of the mean, and so on and so forth. If we go on from that, we would get that different types of standard deviations give us uh, the area bound by different types of standard deviations give us increasing amounts of the population, which gives us increasing confidence as to how much amount of our data will lie within our desired range. And we also go on to see that for in the lectures, that for scientific discoveries, people would often want smaller um, sort of deviations or errors, which would be around 5 standard deviations. So the data should be confined in a small space, and the data should also be within 5 standard deviations of the mean. And here is where we come across the special concept called standard error. So standard deviation and standard error, what would be the major difference between it? Standard deviation would be something which would be the deviation of the samples so both of them almost signify the deviation from the mean. Standard deviation signifies the deviation of samples from the mean, and standard error signifies the deviation of the samples from the mean of means. So that is basically the difference between standard deviation and standard mean. So standard deviation eventually tells us about one sample, whereas standard error tells us about a cohort of samples or a niche of samples. And standard deviation and standard error, if you see, both of them are signified by one single formula, sigma by root of n, where n signifies the number of samples in the number of samples. Yes. And coming back to z, so how many often we kind of see that these kind of numbers which are with the standard deviation, these kind of numbers are multiplied with the one standard deviation, 1.64 or 2.58 or 3 standard deviations. So this particular multiplier value is assigned in statistics, this particular multiplier of standard deviation is called the z value. And z value eventually tells us, like, scientists have, our mathematicians, for many years they have experimented around these kind of z values in various graphs, and they have plotted something called as a z table. So what the z table eventually tells us that how much amount of area out, if the area of the whole normal curve is considered as 1, how much amount of the total area would be bound under a certain z value. By the certain z value, they mean that how much of z into standard deviations. So how much amount of area is bound within z standard deviations. That is eventually what the um, z value tells us. So if we try to plot the value of z value along the z table, we would eventually get the area in decimals. If you multiply that by 100, you would be getting the percentage of area bound by the particular z value standard deviations. And we also come to know about, like, we also come to solve more examples about standard error, and why we have something called as error bars. So you would have come across many graphs where you see this kind of error bars, and which where people would say that it's pretty much data is pretty much significant if the error bars don't overlap with each other, and the data is not kind of significant if the error bars kind of overlap with each other. So why do we need to plot these error bars? So any scientific measurement, if let's just say, in any scientific measurement, if you're going to plot only this bar graphs without the error bars, we are eventually going to say that it's the mean of several other observations that we say. So let's just say this is like quantity as 5 with no units. So what we are going to say eventually is, what we are trying to eventually say is, out of n number of observations, we get a mean of 5. So we are just trying to plot this as 5. But we also saw something about reproducibility in the last lecture. Whereas when we try to perform an experiment, we get a value, and if another scientist tries to perform the same experiment under the same conditions, he should also try to get the same value. But many a times he may not get the exact same value, but some value which is closely related to it. So at that point in the last week's lectures, we stopped with the open-ended key question. If those kinds of small variations are there, what should be done to the data? Should that data be acceptable, or should the previous data be rejected and a new value should be accepted? So this is where error bars come in. Error bars signify under what range under values are under what range should people consider to be a part of that original value with a certain amount of confidence. That is basically what error bars signify. So let's just say error bars, and error bars can be plotted in different ways as well. Many people plot standard deviation as error bars, and if you might have seen in some graphs, the standard deviation is plotted as error bars, and certain other plots, standard error is being plotted as certain error bars. And different types of error bars is used to signify different levels of confidence. For example, if one standard deviation is being plotted as error bars, let's just say I'll just type it in the chat box. So I mean, we consider a value as 5 plus or minus 0.05. So 5 here would be the mean, and 0.05 here would be the single standard deviation bound by the mean. So it means that any value, if any other person tries to perform that experiment, he or she or they can get a value which is lesser than 5.05, or which is greater than 4.95. So this kind of range is fixed for getting certain values from that experiment, and only that range is acceptable with a certain confidence interval. And once again, the confidence this is where the amount of standard deviation, the um, z value of standard deviation comes in. If we have increasing amounts of z value, it just means that more number of data variables are enclosed within that curve, are bound by that standard deviation, and it can be said with more confidence that a certain value is being can be taken under our range. And here an important other parameter called t value also comes in. So what is the difference between z value and t value? So in some experiments we come to know in the central limit theorem that around 25 observations are like considered to be, uh, a good sample to be considered for the central limit theorem. But in not all tests we can get 25 observations out of any given experiment. So let's just say in some experiments we can get 26 observations, or in some experiments we can get only 10 observations. So what do we do in that case? So in that case, we resort to a value called t value. T value is also sometimes it's almost um, similar to z value, and t value also has a similar formula compared to z value. But the only difference is that um, it's just considered the nomenclature is kind of different compared to z value. So Nikhil Chapra is asking, so does this 0.05 plus 0.05 is that bar? I kind of don't understand it. So do you mean that range?
Sir, sir, in this example which you have given,
Yeah.
uh, uh, these bars which are shown, it was I asked this question slightly before some time back when you were telling about bars. My question is, sir, these bars which are shown, an example which you have given, does this indicate that upper limit and lower limit of that bar, orange bar which you said, right?
Yes, yes.
So let's say the first bar chart which is there, its average value is 5, then that upper limit will be 5.05,
Yes.
and yeah, so okay, okay, clear.
Yeah. So yeah, so I can just type out the another range as well. So it will be 4.95, 5.05. Yeah. So the error bars just signify that any value within this range of 4.95 to 5.05 is accepted for that bar if anyone else tries to reproduce the same experiment, same form of experiment. Yeah,
Clear, sir, thank you.
sure, sure, sure. And yeah, and so this is sort of the comprehensive part of whatever was taught this week: central limit theorem, error bars, and standard error. Another topic was also started this week, and only a part of this topic was taught this week. So concepts might not be clear to many of them. So I will teach this concept in the next week as well. But for now, I'll just give a small introduction as to what measurement of proportion just means. So measurement of proportion just means that if we have several groups of population of a certain species we have populations. If we try to group that population into certain species or certain subgroups, how much amount of that group represents the total population? So that is what is means as measurement of proportion. And by, for example, we can say that, yeah, a famous example which was said, which was continued from week 2 of the lectures, was the case of the black moth and the white moth. So white moth was initially more popular in England, and due to industrial revolution, the barks of the trees became more black, and as a result, white moths were more visible to the naked eye, and they were easily subjected as prey by birds. So to combat this, moths were naturally selected by evolution to be black in color, so that they can easily camouflage with the barks and they can escape the predation of the birds. And so, upon a certain point of time, so why this measurement of proportion is needed? That was actually discussed in this final part of the lecture. Measurement of proportion is required for a scientist or a mathematician or any other um, or any other scientist in scientific realm to study change. So science in the modern times, it follows the scientific materialism, which involves the study of change. So we need to study change over a period of time. And a key component of studying these kind of changes is studying how a particular component changes over time, whether it decreases or increases. So let's just take this example. So before the start of industrial revolution, white moths would be obviously more in number. So let's just say 95% of all the like 95% of all the moths were white in color. So 0.95%, right? 0.95 1 and 0.95 is would be the measurement of proportion of the white moths, because white moths represent 95% of the moths of the total population. So you just take the number of white moths and divide it by the every 100 moth. So you'll get the measurement of proportion in that case. Now, as n is the sample size in this case. So that's how measurement of proportion was came into use. By chance, if after industrial revolution, if any scientist wants to study moths and he finds out that, let's just say, the number of white moths have gone down to 40%, 0.95 to 0.4, then he can say that some changes happened, and whatever surroundings were there, whatever um, things were happening around the moths were the cause of this decline of population. So in that point, through measurement of proportion, he could make a scientific observation and give out a study which will benefit natural selection, or which will benefit the, in the time it benefited the field of evolution. And with every study you would need this kind of measurement of proportion to study kind of. Another example I would say is the, let's just say, um, we could consider this maybe playing time by people. How much time do people spend playing or working out or whatever it is before and after the use of smartphones? So any person, any scientist who tries to study this behavioral change, what would he do? He would just conduct a survey of people who used to work out before the smartphones. He would also try to log in the frequency of time in which they try to work out before the invention of smartphones. Let's just say smartphones came into existence around 2008 or 2009 with the invention of iPhones and all the stuff. So before that, people might, work out a lot, go out, and after that, maybe because of the versatility of smartphones, people were subjected to more work, either a desk job or any other or they can also be addicted to apps. Scrolling through apps, or their work might have been made easier, leading them to work more using smartphones, such that their work time, the actual play time or working out time, would have been reduced. So this measurement of proportion comes into play in this case, and the any person who tries to study this behavioral change would compare and contrast the two time periods and see that say that because of this thing, maybe this reduced or this increased, brain activity increased or something like that. So this is a measurement of proportion helps people into finding key answers to questions. And we also went over binomial distribution and over like as a very brief concept. So one small change that we would be observing between binomial distribution and a normal distribution would be that normal distribution generally occurs for a continuous amount of data, whereas binomial distribution occurs for a very discrete data in most cases. And through binomial distribution, or Bernoulli's distribution as the lecturer says, we also come across this samples and data and how you can get mean and variance of a particular amount of proportion. And we come across this important parameter in this measurement of proportion called measured proportion, which is signified by p cap. P cap is nothing but x divided by n, where n is the number of samples of a particular um, number of samples, and x is the quantity of the sample. Yeah. So this is the brief recap we had over the lectures of week 6, covering central limit theorem. And I think for many people it would be a bit tough to kind of because for the past 5 weeks it was entirely theoretical and history based, while this week it was there was a lot of mathematical formulae. Um, People need not worry. Like next week, we can discuss many math-based problems and we can uh, discuss the answers. And yeah, so I'll leave the room for another 5 minutes maybe, and around 3:30 we'll start discussing the answers for week 5. Following which we'll discuss some problems with respect to week 6. So if you guys have any doubts with respect to week 6, maybe you can uh, raise your hand or open your mic. Yeah, we'll at 3:30 we will start the next problem solving assessment session.
Sir, can you please repeat again that P? I don't know how it is called as measure of proportion P cap or what x by n, which is equal to x by n. Can you please explain that concept again?
Sorry, can you repeat? I couldn't hear your question.
Uh, P cap major proportion P cap
Oh,
equal
okay.
to x by n, where x is mean, right, and n is the total number of observations I have conducted, right? Am I
Yeah, yeah,
correct?
yeah. Yeah, number of samples.
Number of samples, huh? Observations means correct.
Yes.
Okay.
Yeah. So let's just say that there are like 10 samples.
Hmm.
So we'll just say 10 samples, and we will give a sort of variable to each kind of groups in the samples. So let's just take this white moth and black moth for example. So out of this maybe let's just say 5 moths are white, 5 moths are black in a sample of 10 moths. So we have 5, that would be because let x be number of uh, black moths. So x would be 5 and n would be the number of total number of moths in the sample 10. So the measured proportion p cap will be 5 by 10, which is 0.5.
Okay.
Yeah. So measured proportion, if you just multiply the measured proportion by 100, it would just give you the percentage of the
Percent.
group in the total species.
Okay. Clear. Clear.
Yeah, Rosalind, you can go ahead.
Yes, sir. Can you please once again explain about the variance and the error?
Okay. So yeah, variance is so. Once again, I'll just go back to the slides. Yeah, yeah. So last week also we in the lectures we started about statistical components such as mean and variance. So variance and standard deviation are just different measures of how a certain quantity varies from a mean of a given sample. So let's just say we have kind of 10 samples, and let's just say the mean of all the samples is around 50. So let's just say that there is another quantity in that 10 samples which has a value of 60. Definitely it's not equal to the mean. It's varying with the mean with around 10. So that that's what typically standard deviation means. So in a sample of 10 we have a certain mean, and but all the quantities won't have the mean as that value. Some of them might deviate with some extent. How much it is varying from that extent, how much is it varying from the mean, that is what gives us standard deviation. Standard deviation just gives us an idea about the probable range within which all the any value can be accepted as being part of the curve. And standard deviation is given by this formula. So sorry, yeah, standard deviation is given by this formula, except that sigma won't come. We will be having a mean, and then our sample kind of uh, our data divided by the square root of n.
Yeah, and sir, standard error. So, in fact, standard deviation is to an extent clear that it is how much is it actually differing from the true mean, up
Yeah,
and below, whatever. So that is clear.
yeah.
No, we have understood the concept. But what is error? Error is my capability to measure.
No, not like your human error doesn't come into this place. Standard error and standard deviation almost have the same formulae, but the values will differ just because of one major difference in standard deviation and standard error. Standard deviation tells us only about one particular component. So let's just say we have 10 samples, and we are going to measure the standard deviation of the sixth component maybe. So it just tells us how much the sixth component varies from the mean. Standard error tells us about the general variation of the whole, all the data in the all the 10 samples in the variation. What would be the general variation of If you take any data at any point in the sample, how much can you expect it to vary from the mean? That is what standard error tells you.
Can we call it as maybe it is a standard deviation of a larger cohort?
Exactly, exactly,
Okay,
exactly,
okay,
exactly.
okay, okay, okay.
Yeah. Gupta, you can go ahead.
Yes, sir. Sir, there is some confusion regarding sample and observations. So, are we talking about the number of observation within the sample, or we are saying single observation as a sample?
So no, sample is so see, a population just means that it can be like it will be a larger data set. So eventually what you cannot do is measure all the properties of the population at any time point. So what you do is you take a smaller part of the population. Yeah.
Yes, sir.
Okay, let's just say that we are in, let's just say that I'm in some factory. So my job is to make sure I'm in a bottle making factory. So my job would be to make sure that all the bottles are manufactured without any leaks. But in a factory, bottles would be produced at a much larger rate. So I can't keep checking, like I can't take any bottle, turn it around and then check, if every bottle is fine and there are no leaks in any bottle. What I can do is I can take two, three bottles from every batch of bottles made. I can just fill those two, three bottles with water and check if there are any leaks. If there are no leaks in them, I can confidently say, with a certain amount of confidence, I can say that, all the bottles are leak proof. So that is the main concept of sample. So you just take
Uh,
some portion from the uh, total amount of population to study the whole population.
Suppose I have 1,000 items
Okay.
and I have collected 10 samples. Each one is having 10 number of observations. So, what does it mean?
So, okay. So yeah. Okay. You have 10 samples and each uh, object in the sample has 10 different observations.
Yes, sir.
So, okay, so in that case, what you would do is to represent that particular sample, you would be taking a mean of that sample. I mean, you would be representing that particular sample with a mean and standard deviation.
Because in the central limit theorem, we just observe the samples, number of samples. So
Yes.
we take the samples mean,
Yeah.
not a single sample, but we take the samples means.
So,
So here I think the n is number of samples, sir. I think, sir.
no, that's what. So, no, so you are actually correct. So, in 10 samples, if you have 10 samples and all of them are 10 observations, so you will totally have 100 kind of observations. So, to plot the samples curve, you would actually make use of all the 100 samples as well. But if you want to sort of, you know, uh, take out one particular sample and then call it, you would take the mean of that particular sample only, take the mean and standard deviation of the particular sample only, and then just write it down. So in that case, the rest nine samples as rule would not be any, there would be any rule for the rest nine samples. For plotting this normal curve, what you're saying is correct. We would take all the 10 observations from all the 10 samples and then plot the standard error and mean.
It means we are not taking the samples mean. Suppose I have taken 10 samples, and
Okay.
for each samples I am taking the mean. So this central limit theorem says I will take the means curve. So
Yeah,
that
yeah.
will be the normal curve. That will
So,
be the normal curve.
so central limit says that you can take any 30 samples. So let's just say we have like 100, like you said, we have 1,000 objects. Okay. So you can take any 30 objects as samples. Not all those samples will have the same mean, and that's when you kind of plot this mean curve.
Because if we are taking only single sample of say 10 number of items, and if I am taking its mean, so it will be a single mean.
Yeah, it'll just
It
be a
will be a single mean, sir.
it'll be a single point in this curve.
Yes, yes, sir. So
Yeah,
that's why I am
that's why they're
asking
taking.
about in this formula we have written z equals to x bar upon mu upon sigma upon root n. So n is what, sir? Exactly. N is items within a sample or number of samples.
Number of samples.
Yes, sir. Thank you, sir. Thank you.
Yeah. So okay, so we are already at almost 3:40. So we'll just go over the problems of week 5, and then we will come back to discuss some more problems in week 6, and then we'll have doubt sessions in between. So if anyone has any doubts in between, like they can raise their hands, and as always in between each question as well, I will also be allowing some room for question and answer and discussions. Yeah. So let's go right away. So yeah. So the term falsifiability means, so they are given four options, sorry. The possibility that a theory can be proved false through observations or experiments. The belief that all ideas in science are false. The belief that science does not know everything. The idea that one can fabricate false data to establish a theory. Sonu is saying one. Yeah, Nikhil is also saying A. So yeah, it means that a theory. So falsifiability just means that there should be any theory at any point, like if it is being in use, if someone comes up with a better experiment to disprove that theory, that theory will be deemed false. So any theory will always have a falsifiability criteria, and it can be proved false at any point of time through observations and experiment if it does not fit into the data that we acquire at any point of time. So that is what falsifiability means. And we'll go over why the other options are wrong as well. So the belief that all ideas in science are false, none of the scientific materialists say like that. In fact, they would want to go with any theorem at any point of time. Yes, actually, there should be a test which can prove it wrong, condition which proves it false. Like the test might not be in a reach during that time due to infrastructure or any non-developments in science. But in future, if we can get any instrument which could prove it false, which we could use to prove it false, it can be proved false. That is what basically falsifiability means. There's meaning that there should also always be a scope for proving it to be false, and that's why certain idealistic principles like the presence of ghosts or heavenly objects or something like that, those are not taken into consideration by scientists because you can never come up with an instrument or any other technological advancement to prove it false. Those are not falsifiable. Does anyone have any doubts with it? The belief that science does not know everything does not fall into this question per se. The idea that one can fabricate false data to establish a theory, um, again, does not fall into this question, and it's a very bad idea as well. Exactly. Nikhil's example is for everyone. You can open the chat box and view at Nikhil's explanation as well. So Nikhil is entirely right. Newton's theory can be proved false with Einstein. Same goes for Einstein. Yeah. So I'll move on to the next question. Okay. One has proposed a hypothesis A and has found an experimentally testable prediction B. So what would hold true in this case? I think this question we discussed it in last class. Yeah, B is Akshata is correct. So B is found to be false, then the hypothesis A is also false. Yeah. I'll move on to the next question. So, suppose a person has, I think this also we have covered in last week. Suppose a person has died and a scientist forms a hypothesis about the cause of her death. It should be based on, uh, it's a multiple select question. So, there are four options: horoscope, circumstances, material processes, and sin in previous birth. So, what could be the possible answer for this question? Yeah, B and C. So we'll go over like since it's a multiple select question, we'll just go over all the options. For horoscope, fate had a role to play, completely idealistic principles. So you can't, for framing a hypothesis, you must always require a materialistic principle. So this option will not be valid. The possibility that she committed a sin in her previous birth, once again, you can't frame any non-materialistic things. Material processes and phenomena that can cause death of a person, absolutely correct. And the facts and the circumstances of her death obtained through observation. So you make observations and you make observations of material objects in your vicinity and then make a conclusion through it. So options B and C will be correct. Does anyone have any doubts with this? I'll wait till 3:45 before we go to the next question. So any hypothesis, like in last week's lecture, we would have seen that in if to frame any kind of hypothesis, we would need to have material processes. It should involve only material processes and material phenomena. It can't involve idealistic principles. Okay, if no one has any doubts, we'll go on to the next question. So the necessary features of a scientific hypothesis are, so we are given four options. They should satisfy the observational facts about the phenomenon. One must have arrived at the hypothesis through deep thought. It should be falsifiable. It should have experimentally or observationally testable predictions. So I think this question also we discussed it last time. Like it was a question which would need some thinking. Yeah. So just like the previous question we discussed, any, I mean, in fact, this comes from all the questions that we have discussed before. So any hypothesis, as per Karl Popper, it should be falsifiable. There must be some falsifiability criteria for it to be proven not true, and then should be experimentally and observationally tested, and should have the capacity to be repeated once again, and it should be first of all observational. It should yeah. So there are three valid options to this question, and the last option, one must have arrived at the hypothesis through deep thought. So thought is something which is like uh, pretty idealistic, and it does not, and any hypothesis must only be material of the materialistic form. So this option would not be valid for that reason. Yeah. So, any doubts from this question? If not, I'll go to the next question. A postulate is a self-evident truth, a scientific guess about a law of nature, a mathematical theorem, an empirical observation. Yeah, B is correct. A scientific guess about a law of nature. Self-evident truth can also be, like postulate can also be a self-evident truth, but for some cases, like you just so in the difference between postulate and self-evident truth is that postulate is always assumed, that therefore that's why it's only a scientific guess. And I think it's just a theoretical question, so I'll move on to the next question. A null hypothesis is not a hypothesis. Hypothesis about Yeah, so Vijay, your answer is self-evident truth, but so the truth part is correct, but it's not always self-evident. So what you basically do with the postulate is you just assume it to be true. Yeah, so it would not be self-evident at all times. Okay. So next question, a null hypothesis is not a hypothesis about the character of zero, a hypothesis about vacuum energy, and an opposite statement of the alternate hypothesis. Yeah, D would be the correct answer. All the three options are kind of, yeah. Basically, a null hypothesis says that something is not the cause of a certain effect, and alternate hypothesis would say that something is the cause of the effect, and we would try to perform some experiment to invalidate the null hypothesis. And if we are successful in invalidating the null hypothesis, we automatically assume the alternate hypothesis. So both the null and alternate hypothesis would be polar opposites of each other. So reproducibility of a scientific result implies four options. The work should result in an industry which will produce the item in large numbers. Experimental animal should reproduce. Another scientist repeating the experiment should get the same result, and the experimenter should be able to memorize the result and reproduce it in an exam. Yeah, I think there's only one option correct in this. So reproducibility just so as I said in the last week's recap and in today's lecture recap. So reproducibility just means that if you can do some experiments in one point of time with the same certain conditions and you get a result, another person doing the same experiment with the same conditions in a different point of time should get the same result. So that's when, so upon reproducing the result, only can you start begin to have more confidence on the scientific result. Next question, the criterion of falsifiability was proposed by, this is also a very theoretical question, Karl Popper. So Karl Popper listed down the theory that every hypothesis must be falsifiable. Yeah. Simplicity of a hypothesis implies its mathematics is easy to do. It makes very few a priori assumptions. It does not require expensive equipment to test. It can be expressed in simple language. Yes, once again, Sonu, you are correct. So it makes very few a priori assumptions. Yeah. And last question, conservatism of hypothesis implies it's based on the available knowledge about the laws of nature. It's proposed by the Conservative Party of England, a statement about energy conservation, an idea coming from a conservative family. So, yeah, the option is A. So, conservatism basically means that whatever hypothesis you're trying to frame, it should be within the body of knowledge that is available to the scientific realm, or or it should be something which is already tested and validated by scientists. Your hypothesis can sometimes it can exist, some part of it can exist out of the body of knowledge as well, but once again, if you have enough testable ramifications about it, then your hypothesis would be valid. So that's what conservatism means. Yeah. So I'll just briefly go over all the options one by one. The term falsifiability means the possibility that a theory can be proved false through observations or experiments. Second question, one has proposed a hypothesis A and has found an experimentally testable prediction B. Then if B is found to be false, then the hypothesis A is also false. Question number 3, suppose a person has died and a scientist forms a hypothesis about the cause of her death. It should be based on the facts on the circumstances of her death obtained through observation and material processes and phenomena that can cause death of a person. Question number 4, the necessary features of a scientific hypothesis are it should satisfy the observational facts about the phenomenon, it should be falsifiable, and it should have experimentally or observationally testable predictions. Fifth question, a postulate is a scientific guess about the law of nature. Sixth question is a null hypothesis is an opposite statement of the alternative hypothesis. Question number 7. Reproducibility of a scientific result implies another scientist repeating the experiment should get the same result. Question number 8. A criterion of falsifiability was proposed by Karl Popper. Simplicity of a hypothesis implies it makes very few a priori assumptions. Conservatism of a hypothesis implies it is based on the available knowledge about the laws of nature. So that's all the 10 questions were there for last week's assignment. So I'll leave the room for another 5 minutes till 4 o'clock. If anyone has any doubts about last week's assignment, we can discuss it now. Else we will move on to the discussion of this week 6's assignment questions. Sorry, I kind of left the call like accidentally. Once again, I'll just present my screen. Yeah, so once again, like I said, I'll just leave the room for till 4 o'clock. If anyone has any doubts with respect to last week's assignments, we can discuss it now. Else, we can move on to the next question, next week 6's assignment questions. Okay, if no one has any hello, okay, if no one has any doubts for now, we will move on to week 6's assignment questions. So I won't be discussing actually all the assignments in the assignment because there were some problems with discussing all the questions from previous weeks. So I am kind of forbid to discuss all the questions. Instead, what I'll do is I'll discuss some of the key problems in this assignment. So that would give you people a sort of an idea as to how to go with these questions and what kind of answers will be kind of accepted. Yeah. So we will start with the questions. Yeah. So some kind of questions. So for the first time in this course, you would have seen that some kind of questions are kind of having a box just asking you to type an answer. So these are numerically answered type questions. So in these questions, people should type any value that you get as an answer, and if your value, so they would NPTEL team would also have a key with them, and that key will not have the exact value as an answer. Instead, it will have a particular range as an answer, maybe plus or minus 1 or plus or minus 2 of whatever answer, whatever would be the correct answer. So if you cannot get the precise answer for any question, it is completely all right. Just try to find any close answer to whatever would be the correct answer as possible. Yeah. So we'll move on to the first question. So suppose you have measured the concentration of a substance in a solution by taking 25 samples from different parts of a liquid and have obtained the following data. The figures are in ppm. So they have given data for 25 samples and most of them lie between 1.4 to 1.5 to 1.6. Or yeah, if you are to report the result in a paper, how will you specify the measured value? Round to four places, round to four decimal places. Yeah. So Sonu is saying 1.548, Gupta is saying 1.5096. So we can try maybe, I think if I have a calculator app here. Okay, many people have, okay, I don't have a calculator app in me, actually, I can't use it apparently. So many people have different answers as well. So like I'll also like work like now, I can just take my phone and then I'll just put, uh, calculate all the values that we get. So, but all of you kind of get how to like solve the answer. So we have 25 values, and to represent all the 25 values, generally what you will do is, but you will measure the mean of all the 25 samples, and then you just give it. So mean would be the sum of all the values divided by the total number of samples. So I just start calculating it right now in my phone. So we have 1.58 plus 1.56, 1.56 plus 1.58 plus 1.49 plus 1.48 plus 1.62 plus 1.57 plus 1.46 plus 1.51 plus 1.54 plus 1.47 plus 1.44 plus 1.54 plus 1.46 plus 1.56 plus 1.43 plus 1.51 plus 1.43 plus 1.52 plus 1.55 plus 1.46 plus 1.44 plus 1.57 plus 1.58 plus 1.48. Yeah. So we get the total as 36.25. 36.25 is total would be 36.25. I am typing the answer in the chat box as well. So total is 36.25, and we have a number of samples n as 25. So mean would be the total 36.25 divided by 25. Total is 37.74. But I am getting 36.25 only. 1.56, 1.56, 1.56, 1.58, 49, 48, 62, 57, 46, 51, 54, 47, 44, 54, 46, 56, 43, 51, 51, 43, 52, 55, 46, 44, 57, 58, 48. Yeah, I'm getting 36.25 only. Yeah. Vijay, you might want to check your calculation because that might give you a wrong answer totally. So I've got 36.25 as a total, and I'll just divide it by 25. Okay, so everyone is getting 37.7399. 37.74 is correct. Okay. 1.736. Okay, many people are getting 37.74 as well. So okay, I mean Gupta, like 37.7399, like all the decimals have only two decimal places, so I don't know how you got 7399. But 37.74, maybe like, yeah, maybe I might have missed one value as well. So yeah, so okay, let's just go with 37.74 as well. 37.7. Okay, so for simplicity purposes, like we'll just go with 37.74 divided by 25. It should be equal to like 1.5096. Yeah, and so yeah, so mean is actually 1.5096. So NPTEL would automatically upgrade, like check in the range of whatever answers you're typing. So any answer from 1.5095 to 1.5097 would be considered as a right answer. I don't know what happened. I think I missed a value somewhere here. Yeah, I'm kind of sorry for that. Yeah, I've done with Excel, mean is 1.5096. Yeah, I mean, like many people are getting 1.5096 only, I think. So yeah, maybe if you put it in Excel or Python, I think you might not miss any values. So yeah, if the answer is 1.5096, then any value between, if you type any value, if you miss some values and if you kind of forget any value, you will still be accepted if your value falls within 1.5095 or 1.5097. Yeah. Okay then. So if nobody has any doubts with it, we'll move on to the next question. Error bars, plus or minus. So it's the same thing. So error bar just means that you have to find the standard error between plus or minus standard error values, and that as well. So the value will be 0.0108. So any value between these two values, 0.0106 and 0.0110, would be considered as a correct answer. Yeah.
Sir, sorry, but how to calculate this? Just you can, even if you tell the process, that is okay, simple.
So by standard error, you just calculate the, so you just use the standard deviation formula.
Okay, okay. Yes, okay.
Yeah.
X bar minus means x minus
Yeah.
average divided square.
The whole square divided by
Yeah, and then
n.
divide by sample and then square root. Yeah,
Yeah, that would
clear.
be the, yeah, whole square divided by n would be the variance. You divide that, you'd get standard deviation.
Yeah, but this second value, how is it obtained? This 0.0106, I could understand. The second, is it plus or minus or what?
It's
This,
plus or minus. It's plus or minus.
okay. No, no. With the formula of variance, we will get one value, right?
Yeah.
Yeah, and in the answer, I see two values, sir.
No, no, no. So that is the range of values. So
Oh, okay, 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 okay. So mean minus, mean plus.
yes, yes, yes.
Okay, clear. Thank you, sir.
No, okay, no, not exact. No, sorry, not mean minus mean minus mean plus. So whatever value we get, so whatever calculation we do, it is bound to some form of errors. So maybe we made, maybe depending on calculations and stuff. Uh, Maybe depending on calculations and stuff, it would bound to vary. So to prevent that, what NPTEL people like, typically for this numerically answered type questions, what people are doing is they would give you a range. If your calculated answer falls anywhere within this range, your answer would automatically
Clear, clear. Understood, understood. Yeah, yeah, yeah. Okay.
correct.
So my answer may be 0.0107 and still it will be correct. That is what it means. Yeah, clear.
Yes.
Perfect. Clear, sir. Thanks.
Okay. So Ravindran has raised a hand, but Gupta standard deviation is 0.0536. Piyush is asking, is it variance, is the error bar equal to variance? Ravindran, you can go ahead for now.
Uh, Arjun Mayuresh here. Just one thing. Sorry, I could not attend last 5 minutes. This
Okay, okay.
error bar, no? Why there are two answers, 0.0106 and 0.0110?
So no, no, that's what. So this is the range. So whatever answer you give, if
Yeah.
your calculated answer, if your answer falls within this limit,
Okay.
so
Okay.
you're
Because I got it. I have calculated and I got it 0.0109.
yeah, exactly. So your answer falls below within this limit, right? 0.01
Okay. Right.
6 and
Yeah.
0.
Yeah. Yeah. Yeah.
Yeah.
Okay. Okay.
So it means that if your answer if falls within this two numbers, then your answer.
Yeah. But from our side, it should be single answer, right?
Yeah, yeah, it should be
From
single answer.
candidate
Yeah,
point of view. Okay.
because you
Right.
can type in
Clear.
only one answer in the answer bar, the numerical bar.
Right. Right.
Yeah. But
Yeah.
did you calculate standard error or deviation?
I have calculated standard error. Deviation is into five
And
times of
yeah, I
this.
think it. Yeah, yeah, because it's so for error you take the n, whereas the
Yeah, sigma divided by square root n has given this answer.
exactly, yeah. So it's standard error. Okay, okay then. Yeah, I think many people might get like confused because I think for regarding the square root value, standard error would take in the square root. It would take in two square roots, so it will actually become five times less than whatever answer we are going to get. Yeah, 0.0194 would automatically fall into this answer as well.
But it is also told to put things within four decimal places.
Yeah, so if you can just put like if you get any answer in two decimal places, just add two zeros.
Yeah, yeah.
Yeah, it would, so that does the extra decimal places wouldn't be mattering, right? So if you put just two zeros, it's going to be automatically,
Right, right.
yeah, exactly. Ravindran, you have raised a hand or like, Piyush, yeah, you can go ahead.
What is the difference between variance and error bar?
So variance will not actually be plotted in an error bar. Error bar would normally, so as I said in the recap bar, your error bar will often have the standard error. Hello, can you hear me?
Yes.
Yeah, so standard error is almost similar to the standard deviation except that it takes the whole population into account, whereas variance is the square root of standard deviation, which tells you how much a quantity varies from the given mean in a sample.
Okay.
I guess this like everyone gets the how to solve this question. Okay, if there are no other doubts, I'll go on to the next question. We can always come back to this question to discuss what actually happens in this question in the doubt session. So, a bioscientist only can take 6 observations for his measurement as the restriction of the lifetime of the bacteria he is working with. In his case, how can one define the confidence level? So here they given that he has only 6 observations. So saying he can define the confidence level using t distribution. Yeah, so that is correct. So normally we would have a according to central limit theorem we have a normal curve and we have a z distribution according to which if we have a large number of samples, preferably 25 or above samples, we can say with a particular confidence that uh, we can define the confidence level of any data that we are presenting. But here he has only 6 observations, which is like very less than, the normal amount of observations that we define. So for smaller observations, instead of z distribution, we have something called as a t distribution, and we for the t value we consider the same thing as t z value. The t value is almost similar to z value, but what we do with the value would sometimes be different. Yeah, and it may in compared to a z table where we have only one value for a z value, we have multiple values for a certain t value depending on the confidence interval which we want to present the data in. So depending on the constant confidence value and our t value, we will be given a value, and if that value is greater than the t value that we have, either positively or negatively, we can say with confidence that our data point lies within the uh, given confidence interval. So we can say with confidence that our data is there. Yeah. He can define the confidence level using, so coming to the other options, he can define his own confidence level. Uh, you can, but you have to define your own standard deviation and metrics as well. There is no confidence level for his observation, incorrect. So because t distribution is there, he can define the confidence level using normal distribution, incorrect once again because he does not have that many observations. So does anyone have any doubts with this? Okay, so I'm guessing no one has doubts. So that's all for the question. So these are the different kinds of questions that I saw, like of variability in the question and answer session. So the rest all were just follow-ups of all the questions. So I'll open the session from now to 5 o'clock. 5 o'clock is the end of the session for doubt session. So I'll leave it till then for doubts. We can discuss the questions in detail for the week 6 assignment as well. And if you have any doubts with the week 5 assignment also, we can discuss. Yeah.
Arjun, can I ask doubt? Mayuresh
Yeah.
here.
Yeah. Sure.
Yeah, yeah, Arjun, this question number one, no, which is which is connecting between problem number one to problem number 5, 6.
Yes, yes, yes.
for assignment 6. So there no, they were asking in question number 3 and 4 about the X range to Y range. That is the limits on either side for 95% probability.
Yes, yes.
Yeah. So this 95% probability is nothing but this 2 sigma, no? 2 sigma distribution. That is standard
2.58.
error in, yeah. So standard error into 1.96 times of standard error on either side of the limits
Yes, 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 yes.
for 95%. Yeah.
1.96
So
is
with
for two
that,
standard deviation and 2.58 is for three
yeah,
standard deviation.
yeah. So 2 times, yeah.
Yeah.
Yeah, two sigma. For 2 sigma, it is 1.96, and exactly they also asked it is for 95% probability. So with that, X range and Y range is obtained, some values, okay.
Yeah.
But then the next question, no, that is question number 5.
Uh,
They
can
are,
you just
yeah,
give me like 10 seconds? I'll just switch back to that assignment questions if I have them.
yes, please.
Yeah, sure. Now, which question were you talking
Yeah,
about?
question 3 and 4 first. I'm talking about question number 3 and 4,
Okay.
where the, yeah, where
Define a
the range is
range.
asked, X range and Y range. That is X, that is low limit and high limit.
Okay, okay, okay. Define a range in
Is
which
it
the
not? It's a low limit to high limit.
okay. Define a range in which the true value of the concentration must lie with 95% probability. Uh, I think you can give the lower limit for this one. So I think X
Yeah, so no, yeah,
range.
X is a low limit and Y is a high limit.
Yes, yes, yes. X is the lower.
Yeah, so with that, okay, I obtained that. My question is on question number 5 now. With respect to
Okay,
this,
yeah.
when
So
I'm still seeing,
they have automatically given
this is okay.
the range is X to
Yeah,
Y. So X would automatically
yeah.
be the lower limit and Y would automatically be the higher limit.
High limit. Yeah. Now, if you say no, this X and Y we obtained, and now when I'm going to the question number 5, this question number 5's range is within question number 3 and 4. So, like question number 3 and 4, no, there is a X low limit and high limit. So, this question number 5's limit falls within that subset of this. But still, when I'm calculating that with the confidence level, when I'm calculating with respect to the Z table, I'm getting confidence very low. That is 62% only I'm getting, which actually is a counterintuitive. It should have been more than 95%. Are you getting my point?
Yeah, 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 I'm getting it. I'm getting it.
Yeah.
Again, so
Can I tell you the values so that it will be better?
no, no, can you just give me a second? I'll just go over the question
Yeah.
once more. So, with what confidence level you say that the actual value lies in the range 1.5 to 1.5192 around 2 decimal places?
So for question number 3, it was
Okay,
very
okay.
straightforward. It was,
Yeah, yeah, okay, okay. So okay, I got the question actually. So we discussed that the mean or the mean value whatever we got that's around
yeah,
1.509, right?
yeah, yes,
So let's just take that mean as a reference mean
yeah,
value. So from
exactly.
1.509 to take this 1.5 on one side and 1.51 on one side.
Yeah.
So I think this 1.5 and 1.51
It is exactly
plus,
halfway,
yeah, yeah,
0.0096.
yeah, yeah. Both of them like halfway of 1.5 and 1.5192 would be.
Exactly, exactly. Yeah,
Yeah. So what they are actually asking is how much like what would be the standard deviation?
confidence level. Yeah.
How much standard deviation from our mean 1.5096 would be 1.5 or 1.5192?
That they are asking about the confidence level, is it not? That's a probability.
Yes, yes, yes. So you must
Yeah. So
uh, uh, like so either you can either put it in percentage or you can put it in decimal value.
yeah, but in that case, when I'm calculating the z value, no, for getting this probability, I need a z table.
Yes,
So
yes,
I need
yes.
to find out the z value. So z value comes out to be, I have taken one of the extreme, that is 1.5096.
Okay.
That is the mu minus
Yeah, yeah.
the one extreme 1.5. So
Yes, yes.
1.5096 minus 1.5, which is
Okay.
divided by the standard error, that is 0.0109.
Yeah, that would give you the number of standard
Yeah. So
deviations.
Yeah, so that gives me 0.88 for looking up from the Z table. And from
Okay.
Z table, I'm looking this value, it comes out to be 0.81. 0.81 is the area on the left side of the Z table.
Okay.
Yeah, and then the one minus of this will be the point, area on the right side, 0.19. And then double of that will be the area under the left limit and right limit and left limit.
Yes, yes.
So my question is no. So this comes out to be only 62%. Okay, my question is
Yeah,
now.
yeah. Actually, that would be the correct answer, right? So,
No, but no. My question is no. It is counterintuitive to me. Why
okay.
why why it is so? Because question number 3 and 4 has given 95% probability with a specific range, and that range was. Can
Yeah.
I
So
spell that numbers also?
no, no. I myself will
Yeah,
just tell you
yeah,
one example. So let's just say
yeah,
95% probability. You can say the lower
yeah,
limit is around 1.45.
yeah,
Okay, 1.45 or 1.55. Lower
yeah,
limit can be 1.45 or higher limit can be 1.55. That
yeah,
say that lower limit and upper limit with 95% probability. But here
yeah,
they have narrowed down the range.
yeah,
So wouldn't the confidence, so just think of it in this way. So you have a higher range, you have a higher confidence interval.
yeah,
So the range is
yeah.
narrowed down. So wouldn't your confidence also narrow down, come down?
Okay, okay, okay. So when the
And
limit becomes
so
subset, yeah,
exactly. So you got your standard deviation. So you did that math, like 1.5,
yeah,
like that. There itself you got your answer actually. So 1.5096 minus 1.5 divided by,
yeah,
you divided by your standard error, right? And then you got
yeah,
0.81, right?
yeah,
Yeah.
yeah,
Normally, you know that one standard deviation is going to give you a confidence level of 63.7%.
yeah,
So zero point 0.8 standard deviation, 0.81 standard deviation or 0.88 standard deviation is going to give
yeah.
you something a bit less than that, right? 62% or 61%.
Okay, 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 okay. Clear, yeah, clear.
Yeah.
Thank you. Yeah,
So
yes,
Z
yes.
value is just to pinpoint your answer to one specific value. So you can cross check, you can back calculate or cross check your value using the small hunch.
Yeah, yeah.
I hope
Only
it's
thing is
clear
no.
for you.
Why I was confused because this the right limit of the fifth question is 1.5192, and
Ah,
question number 4 the right limit is just decimal away, third
yeah,
decimal away. But that is going to change such a drastic confidence level. That only was a confusion.
yeah. So yeah, like Z value, like I said, Z value is just to pinpoint your answer to one specific confidence interval. Apart from that, you can just back calculate your answer to this specific particular hunch. You have this values always. One standard deviation is going to be 63.7% of your population.
Yeah,
2.58 and three standard deviations is going to be 95% and 99.7%. So you
yeah,
can always check with these values to see how closer or how farther away are your values. So
right,
that's I mean, for if you are a bit confused, I will say that that's a much more easier way of checking your answer twice.
right, right.
Yeah. But I think that was a good question. It would confuse a lot of people, I will say. Yeah. So does anyone else like, I think by this three, so for the other people in the room, I think by this question we have already discussed like all the first five questions in the next assignment as well. If you people are keeping track of all the questions. So first two questions, I like in the assignment, I think I discussed the first two questions, and Mayuresh kind of discussed the next three questions as well. And if you people are like confused with accessing Z table, you can just, so Z table is a very pretty consistent table. Everywhere the Z table is going to be consistent. Uh, if not, I can provide under Z table like a PDF form of the Z table in the Google groups, the discussion forum. I can try to send it as soon as possible. If you people are struggling to find Z table or T table. Sure, sure. I'll try to, yeah, okay. I'll try to send it as soon as possible. I'll just note it down. Can I also know when is the deadline for this assignment of week 6 from anyone?
Sixth.
6th March, okay. Okay then, okay then. Yeah, Gupta, you can go ahead. Ravindran, are you still, once again, Gupta, so Ravindran, are you still raising your hand? Like, if not, like, I'll just lower your hand if you don't have any doubts. Gupta, you can go ahead, sorry.
Sir, can you once more explain the error bar concept, sir?
Okay,
What
so
exactly formula we are applying here, sir?
okay, so in one of the, sorry, once again, in one of the lectures you might have seen the error bars formula. So we already have the populations. Let's just assume that we have the populations, sorry, population standard deviation sigma. I'll just see if I can write it here. Yeah. So let's just say we have sigma as the population's standard deviation. So the population standard deviation would be like summation of any quantity minus the mean whole square divided by the n, the number of samples, and this you would be rooting it off. So this is the general formula of standard deviation. Yeah, standard deviation. So provided,
Yes.
sorry,
Yes, sir. Yes, sir.
yeah. So this is the general formula of the standard deviation. This we saw in the last week's lectures as well. So standard error of any quantity, so that we represent that as sigma of x. That will just be given as sigma. If we have this is provided when we have the mean standard deviation of the population sigma, once again divided by the square root of the number of samples. So when you kind of like put in this formula, it would become the square root of this will come only to this, and square root of n will become n, because square root of n into square root of n will become n. Yeah. And
Mm.
yeah, and I think you all like for the second question also, like they are asking about error bars, right? So while trying to calculate the standard error, you must take the standard deviation and then divide it by another root of n, which will like divide it by 5 times instead of root of 5 times. Is it clear now?
It means sigma upon root n. We have to calculate this value.
Yes.
So this will be the plus minus error.
Yeah, this will be the plus minus error.
Okay, so it will be a single value.
Yes.
Plus minus. So it may plus value positive or negative.
It will be it will be plus or minus. So
Okay. So if we talk about
because your standard deviation, that will always be in the plus or minus value, because you are rooting it from variance's value will always be positive, but you're taking a square
yeah,
root of your variance, which will give you the standard deviation. So standard deviation, you can't like specify the sign of it, it will be plus or minus, and that's why you're plotting your standard deviation on either side of your mean.
Okay, sir. That means if
Yeah,
let's take a very simple example, we have 25 observations.
yeah,
So n is 25. Okay,
okay,
and let's say the standard deviation is 10.
okay,
Huh? So the error will be 10
2,
divided by square root of 25.
2, yeah,
So it will be 2 plus or minus 2.
Yes.
And so that bar, so error is actually that bar, right? Which is
Yes.
shown on that in that graph on the block. If the average mean was, let's say, 7, and
Yes.
so it will be 5 to 9. That will be that bar, right?
Yes. Yes.
7
Yes.
minus 2, 7 plus 2.
Yes.
Error. Okay, clear, sir.
Yeah. Just like I said, standard deviation just gives you the property of one specific value, whereas standard error gives you property of the whole population in a set of samples.
Okay, okay. Clear, sir. Thank you very much.
Sure.
Okay, right, sir. Thank you, sir.
Yeah, sure, sure. Yeah, and like coming back to Nikhil's thing, like it would be 10, but it will be plus or minus 10, not exactly plus 10, because standard deviation uh, is positive or negative. It deviates from the mean on both sides.
So it's a kind of range. That means the value can vary
Yes.
from minus to
No, no,
plus.
standard deviation's value won't vary. Standard deviation defines the range. Standard deviation is not a range.
No, I am asking about the error, sir. Error value that is range from
Yeah,
plus
yeah, yeah.
minus to
So
plus.
same error also defines the range. It is not a range. Error, your error defines the values, defines the range in which your values can be. Error,
Okay,
error itself, the value of the error will be a singular point. It will be a constant. Range left and right.
okay, sir. Okay, thank you, sir.
Yeah. Yeah, Nikhil. Thanks. So I'll leave the room for another like till 6:40. Then we can end the session if no one has any doubts.
Mayuresh here again.
Yeah, yeah, sure.
Just one thing is, I was just looking at Z table
Yeah,
and the standard no one sigma value, for example, that is
yeah,
68.3%.
yes,
That one sigma, if we take limits, it gives a confidence of 68.3%.
yeah.
Now,
Standard deviation.
from Z table, when I'm searching 0.683, 0.68, that is 0.68, it is mentioned as 0.7517. Z table value is 0.7517. That is 75% confidence. That only I'm having a doubt.
In Z,
Yeah,
trying to search for 0.683 value.
yeah. I'm trying to search for a Z of 0.68. Is it not? Or I
Yeah,
should search
but
for 68%.
no, no,
I
that
should
is the
search
thing.
for.
See, so if one standard deviation is giving you 68.3% confidence, then your Z
Yeah.
value is 1. It's not 0.683.
Oh, Z value should be 1. 1 second. Let me search. Wow. Yeah. Z value at Z value of 1.
Yeah.
What I should search at Z value of 1. Is it possible for you to project one Z table and show? That will be really nice.
Yeah, I can try once again. I just randomly put it.
Yeah, yeah, it's available in the Google. Just
Yeah, yeah, yeah.
because Z of 1 also is giving something like 84% confidence.
Yeah. One second.
84%.
Okay, but I'm searching for Z values which will have 1.
Yeah, Z
Yeah.
of 1,
And
no, like the first column.
yeah, yeah, yeah.
You got the Z table?
Yeah, yeah, yeah. I'll just project it now. Yeah. So can you see the Z table now?
Yeah, yeah, yeah.
Yeah, I'm just trying to search where the one would
Yeah,
fall.
Z. Yeah, the first left column, no, first column.
Yeah, yeah, it will be around.
That's the Z column. So Z is equal to 1 if you see it is 0.8413. So that gives a confidence a probability of 84%. No? There only
No, no,
was
no.
my
So
doubt.
okay, okay. So no, no. So okay. So in the Z table, what you will get is if you have a Z of one to the right.
Yeah.
So here, okay, I'll just scroll this up a bit. So you can see
Okay, I
that.
have to put it two times probably. Yeah, I got
Yeah,
it.
yeah, it's fine. So you have 0.8413 here. So it means
Yeah.
that if Z is 1, the area to the left of this Z curve covers around 0.8643 of your total curve.
Right.
Okay, sorry, 0.8643. Yeah, 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 0.8643. So you do the usual thing. You subtract it by zero point 0.5 and then multiply it by 2.
Clear. I'm clear now. Yeah,
Yeah,
because I
you
was
will get,
double. No. Yeah, yeah,
yeah,
yeah.
you will get
Yeah.
68.26.
Yes, yes, yes. 68.26.
Yeah, so if you subtract it by zero point 0.5, you will erase this
Yeah.
portion to the left
Yeah.
of 0, and then if you multiply it by 2, you will get the portion bound by plus z and minus z.
Yes, yes.
Yeah, I wish I would have a drawing table. Wait, I'll just copy this image and paste it to my PowerPoint. Then I think I can draw.
Yeah. If other people are clear, you can go ahead with other people's questions because some people are raising some doubts. I am clear now.
Yeah, yeah, it's fine. No, no, it's, I think everyone needs to get a grip of this concept as well. So I'll turn on the sharing and then I'll just send my. Sumit Shukla, I can see you are raising your hand, but I'll just come back to you because I think we have 20 minutes, so I can back your doubt. So yeah, so you can all see this Z table once again. Like this is for everyone, I would say. So you can all see the Z table. So Mayuresh's doubt was like in Z table for one standard deviation, we see the Z value as 0.8413. But how could we say that one standard deviation actually gives 68.3% confidence interval? So how you actually use Z table is that whatever Z value is corresponding to whatever Z score, Z value that you have, that actually represents to the area of the curve subtended by to the right of the positive Z value. So if Z is 1, you would say that to the left of Z, it covers almost 84.13 percentage of the total curve of the normal distribution. So how do you actually determine how much is subtended by plus one standard deviation and minus one standard deviation? Yeah. So I'll just mark this as well as minus one standard deviation. So this is the area that we want to calculate. So what you actually do is we have more than 50% of the curve occupied. So you subtract 50%, you subtract 0.5 to give us the area subtended in this specific portion around after 0 till z. So that will give us, I will type it in the chat box. So 0.8413 minus 0.5 equal, it will give us 0.3413. And if you want to find the area between plus z and minus z, you just multiply it by 2. So 0.3413 times 2 gives you 0.6826. And if you multiply it by 100, you will get 68.3. 6.26%. So this will be the total area of the curve subtended by the plus one standard deviation minus one standard deviation above around the mean. So that's how we actually calculate.
And
If it is a negative value, if z value is negative, how do we do it?
So if z value is negative, there is another chart for that. There is another z table for that. Yeah, angle subtended to the left of the z value. So that will give you the value. So there are two different z charts. One will be above 0, one will be below 0. So each one of them will have separate values. Based upon that, you should try to do the math around it and then try to find the angle around it.
No, I'm just asking like how do we do it if it is a negative?
Okay, so there is another z table. I'll try to paste that as well again.
No, because whatever the z table I saw on the Google, it contained from negative to positive value all.
Yeah, yeah. Just give me a second. I'll just paste that as well. Can you all see the new z table now?
Yes.
Yeah, so this is the z table where we have z values in negative form. So we have 0 till minus 3.6, and here once again, like whatever value we get, that is the area of the curve subtended to the left of the z, whatever z value we have. So let's try to do the same thing once again. So for 1 we have 0.1587. So this shaded area would represent 15.87% of the total curve. So if you want to once again subtend the find the angle, like sorry, not the angle, the area between minus z and plus z here. Just put it as z. So you try to find this value and then multiply it by 2. So you know that mean is almost 0, mean is 0.5, and you already have 15.87 around it. So just try to subtract 0.5 minus 0.1587, which will give you almost 3413, 0.3413. And then once again, if you want to find the this is 0.3413 is the area between minus z and 0. So if you want to find the area between minus z and plus z, you will multiply it by 2, which will give you 0.6826. So that is how you use the z values in the negative side and positive side. If you get it in the positive side, you will subtract that value from 0. You will subtract 0.5 from that value. If you get it in the, if you have any value from the negative z value, you will subtract this value from 0.5 and then multiply by 2.
Okay, got it.
Yeah. So yeah, Sumit Shukla, you were raising your hand, I think.
Sir, got my answer, sir.
Oh, okay, okay.
Thank you very much, sir.
Sure.
Basically, this jet chart is a conversion of the whole distribution in area,
Yes, exactly.
and in sigma we calculate in the percentage.
Exactly. If you consider the whole area of the curve as 1, how much proportion of that 1 is whatever area subtended to the left of z? That is what this chart is trying to tell us.
And this 3 sigma is the industrial standard.
5 sigma, oh yeah, 3 sigma is not industrial standard. I do not exactly know. Some companies might go for 4 sigma as well. For scientific discoveries, we need 5 sigma. 3 sigma is suitable for scientific, whatever studies we do in our day-to-day life, whatever experiments we do. That would come under 3 sigma value, which will cover almost 99.7% of the curve.
Correct, sir. And we are not going for 6 sigma.
No, I think 6 sigma. No, I think 6 sigma is just a
concept only.
Yeah, it's just a figurative concept. It will be like very, very rare,
Okay. Thank
very
you, sir.
rare chance of going out of fault. That's what Six Sigma means.
Fine. Thank you, sir.
Yeah. Guys, I'm getting some disturbance, so can you mute your mic if you don't have any doubts?
Mayuresh, once again, just one last out. That
Yeah,
is
sure.
one of the number I'm getting as, for example, 1.4882. And it is told that answer should be in two decimal places. So 1.4882 should be 1.49, is it not? Because
Yes.
third decimal place is more than 5.
Yes.
So it
Yes.
should be 1.49.
1.49. Yes.
Yeah. All these answers which are to be entered, no, will be having some range, right? Just
Yes.
for,
Yes.
yeah.
Every numerical answer. So, some people might have like types of computing all these things. So not everyone will be using the same calculator. That's why we have a range for all these answers. So you people don't worry. If you
Okay, clear.
think your
Thank you.
answer is close to any expected value, just put it in. Okay. Are there any other doubts with respect to this? Okay, then if none of you have any doubts, we will close the session for now. Once again, thank you all. Like it was actually a very interesting session compared to all the five sessions we had. A yeah, Shreya, you can go ahead. Shreya, you have any doubts? Like you have raised your hand. Yeah, it's okay. Even if you guys don't have any doubts, like I mean, if you can't tell your doubts now, you can later put it in your.
Sir, myself Shreya.
Yeah.
Hello.
Yeah, I can hear you. I can hear you, Shreya.
So I have joined later, so can't. I want the recording of this session. Can you please?
Yes. So this lecture is actually recorded. So this lecture is actually recorded. So if you go to
Uh,
NPTEL portal, if you go to the course portal in the NPTEL window, you will
uh,
find a list of if you will find an Excel sheet in the problem solving assessment section. So you will have
oh,
week 1, week 2 till week 12 lectures, right? So
okay.
after that you will have the problem solving assessment sessions tab.
Oh,
So if you access
oh,
the tab, you will have all the video lectures of all the weeks that we have done so far.
okay, sir. Okay, sir. Thank you, sir.
Yeah.
Okay.
Yeah, and this is for everyone else in the call as well. So all the lectures that I have been doing are recorded, and the YouTube links for those recorded sessions have been posted in the problem solving assignment Excel sheet. Sometimes I might take 1 or 2 days to upload the video due to some internet issues, and then I'll post the link. But I can make sure that the video is uploaded by Monday or Tuesday, so that you people can access it before the deadline as well. Uh, yeah, and if you have any doubts, you can either post it in the YouTube comment section, or for a detailed discussion, you can also post it in the Google Groups discussion forum. Yeah, and that's about it, I guess. So thank you all for coming to the class. Um, it was a, I think, fruitful discussion for me and you guys as well. So our next class will be on the next Saturday, 9th March, between 3 p.m. and 5 p.m. Yeah. So thank you, everyone.
Thank you. Bye.
Yeah. Thank you.
Thank you, sir.
Thank you.